All right. So this presentation is going to evolve around 4 different technologies that we use in my lab, and I think have broadly relevance to you today. And so it's fundamentally about using technology, how we can use it in a way to recruit students to write successful grants, but I think very importantly, how we can make ourselves as faculty, how we can make ourselves relevant to the society which we are part of, and how we can see our involvement at universities as a way, as a platform to produce something that other people can use and can lead to jobs, can lead to careers, patents, ways forward, especially the young people who are maybe looking at the future and thinking, where do I fit in? What are the options? And try to turn the narrative into something positive and look at climate change and other huge challenges as a set of opportunities. So that's really what I'm very, very focused on and very, very interested in promoting. So the presentation here is, if I can get this going, there we go, is really looking at these 4 different pillars in my program, and I'm going to go through each of them individually and hopefully give you a sense of how we can use these technologies in my program as a way to entice students to pursue degrees in sustainable crop production. So we're going to start with cold plasma, something you don't see a lot of in entomology departments around the world. I bet we are the only ones that have a cold plasma system. So what this technology, this is a large grant we got last year, where we are now able to ionize atmospheric gas and blow it into water. This completely changes the characteristics of water. I'll show you some data here in a second. And one of the reasons why we were able to put this together is because, where's Neil? Neil, where are you? There he is, sitting here in the front row. Neil and 2 other collaborators at University of Maryland and at University of Minnesota got involved. So we have a fantastic team working on using this technology as a way to really fundamentally change, especially for the organic crop production, how you can look at crop production, because this plasma activated water has some characteristics that makes it extremely interesting. And I'll show you some data here where you can basically just see on these 4 graphs that as the time, which is the x-axis in these graphs, you can see that the pH, the conductivity, and the oxidative reactive potential, all these characteristics of the water completely changes as we treat this water. And when we then take this water and soak seeds into them, guess what? It's like magic. We start seeing that they germinate faster, they germinate more uniformly if we use a certain level of treatment with this cold plasma system. So you can also see on the graph to the right that there is sort of an optimum. If we make it too acidic, then that water is actually going to kill the seeds. But there is some potential here, and we are now seeking more resources to dig further into this and really look at how PAW, this plasma-activated water, can be used, especially as a fertilizer for the organic industry, something we are very, very interested in. So we get faster seed germination, we get more uniform germination. Well, that is very interesting for those of you who are involved in large-scale commercial production, to have more uniform productions, spending less time in the greenhouse, so you have less exposure to pests. The crop can get out the door more quickly, and because it's more uniform, it's much easier to roboticize, which gives you some of you in the audience I know are working for companies that work with greenhouses and with roboticization. And well, this is where PAW actually comes in as a potential collaborator in this whole concept of growing crops more effectively, more uniformly. And definitely also get more vigorous seedlings because it affects their root structure. So also if we are looking at transplants, actually getting them out in the field, they are potentially going to overcome that shock more effectively, which is something we're also going to be looking into. And we see that PAW, this use of water, can play all sorts of roles in pest management. The characteristics of the water gives us a lot of hope, and there's a lot of studies already out there that is being used in control of fungal diseases and other things which are huge problems in greenhouse production systems. So this is very, very unique and very, very interesting, and certainly something we would be very interested in talking more with you about if you are interested. And we do submit a newsletter if this is something you would like to be receive news on and how we are moving forward. Please let me know, and we can add you to that list. So that's sort of the first pillar in my program. Another is our advanced lighting, and I see Heiner here sitting in front row. Heiner is somebody who has definitely helped us out and been a fantastic collaborator. It's something that I think people that are older than I probably looked at long time ago, and then there were some technological constraints, some financial constraints in that technology. Now with LEDs, this is getting a renaissance. It's definitely changing the landscape. And because more and more food production is moving inside, and because there's so much automation and so many electrical installations already present in the greenhouses, there's something to work with here. So we are looking into how we can use this. And we are starting at some smaller levels where we look at different LEDs, and we look at how we can affect pest populations, both directly, how we can actually suppress some of them by exposing them to harmful UV light. And there, of course, we want to be careful not using wavelengths that also harm the plants. So we have to look at different ranges of UV light so that we can not negatively affect the crop, but have an ability to control our pests with it. So what we are looking at is, in one of the studies that one of my students is doing, we're looking at Western flower thrips, and we are exposing light either from the top or from the bottom, and we're looking at different wavelengths. So what he has done is he has exposed these tomato plants that were experimentally infested with Western flower thrips, and then exposed them to light. And what he then does, he has exposed them at night versus during the day, and also from the top, so on the adaxial side or on the abaxial exposure, to see whether that made a difference. Because it would be very interesting to know more about whether the circadian rhythm of plants would in some way impact how they respond to these different light conditions. And then what he does is he does some staining of the leaves, and we count the number of non-hatched eggs that are still left in the leaves, and he counts the nymphs that come out. So it's basically some bean counting, and what he comes out with is, if these plants are exposed to only 4 hours of a certain wavelength of UV light, then we can, as you can see on this graph, you can see that irrespectively of whether it's done at night or whether it's done during the day, we can see that there's a marked reduction in Western flower thrips that emerge from these plants. So we are now actually proposing this as a way to directly go back to one of the old school physical pest management practices, using light as a way to control one of the toughest pests that are out there in greenhouse crop systems. So this is quite exciting for us. We are also working with spider mites, and I should say that both spider mites and Western flower thrips share one particular, they share common several things, but one of the issues is that they are both hidden. They are both very difficult to target when you spray with contact insecticides. And for the organic industry, of course, there are not many contact insecticides available in the first place. So what we're really trying to do is come up with something that could potentially synergize the somewhat reduced effect of low risk pesticides. And that is particularly interesting for spider mites, because those of you who deal with spider mites know that they typically are prevalent in the bottom part of crops and on the abaxial side, on the underside of leaves. And that's why they're very difficult to control. And they're not being effectively targeted with systemic insecticides. So you have to hit them with something, but they're hidden in the bottom side on a dense canopy. So we are thinking, what if we could move them? What if we could manipulate them? What if we could use light to push them upwards into the canopy, up to the top side and in the upper portion of the canopy? And so we've done a whole bunch of experiments, and I'm just going to share some very simple ones. So in one of these experiments, we first did some screening of some different light conditions. So on this table to the right, you can see that we were recording their movement, and we basically wanted to find out how much they moved around. And the idea was the more they move, the more they're trying to get away from the light. So we were just screening through a whole bunch of light systems, and this was in an open petri dish, so very experimental conditions. And you can say, well, what does that have to do with what you're talking about? Well, you will see the relevance here in a second. Because when we then did the same experiments, but provided them with a little bit of a shelter, then in most of the cases, what happened was they went into the shelter. They did not go outside, so they were hiding and protecting themselves from the light that they were exposed to. But there were some light sources where they actually spent more time, disproportionately more time, outside the shelter. So that gave us an idea that they were actually being attracted to the light. So with this simple setup, we were able to demonstrate that some light sources were attractive and some were repellent. And we thought, well, and this is what you can see here, that blue light, the certain wavelength of blue light that we used, they spent much less than what predicted by random, which is what that line at 50%, what that shows. That was because they were not spending it inside the shelter. They were doing it outside, while the UVC, which of course is very, very harmful to them, they immediately ran in under the shelter and kept themselves, tried to protect themselves. So no surprise there. But what that meant was that we could essentially come up with attract and repel system, where we push them up that way. So what we have just finished, actually just a couple of days ago, is we made a little experiment where we just for 10 minutes, we had infested some tomato plants with 50 spider mites, all of them, and we had 3 treatments. We had no spraying. We had some that we just sprayed with a Botanicard, which is a Bulgaria organic pesticide, and then we had another group of plants where we sprayed them with the same insecticide or miticide, but 10 minutes prior, we had exposed them to this light regime that we have found out was very attractive, repellent. And guess what? When we then came back 5 days later, we were able to demonstrate that we could get very, very strong control. So this just demonstrates that all technology, if it's thought of in a clever way, in an innovative way, there are definitely ways that we can use behavioral ecology. There are ways we can use technologies to really become smarter and do things. We don't need to necessarily rely on the hard chemicals. We can get the job done if we are just willing to think outside the box and use all the technologies at our disposal. So there's a lot of light engineering. I'm in entomology, but there's a lot of engineering that goes into this. There's a lot of ways that we can work with the pesticide companies about improving not only organic but also conventional pesticides. There's definitely whole ways that this could be integrated into programs. So this is something we are very interested in taking further, and not just write papers about, but actually develop solutions and use some of the resources that are available at our university and beyond to see how we can take this further. So that finishes up my advanced lighting component. Now we're going to move to what we do with robotic spraying, because we also have a roboticized spray rig that we can use. And just to, I know most of you work in greenhouses, but just we have some of the same issues in greenhouses, but definitely out in field crops, we have this problem that when we go out and spray, irrespectively of how much we spray, the spray volume that we are using, which is what this shotgun shot shows here, is you think that as you use more water, you're going to get better spray coverage. Well, the problem, this is actually real data with commercial spray rigs from strawberry. So we're not talking big crops here. We're not talking corn or soybean or something. This is a small crop. But what you can see here is that there's a very, very poor correlation. And what that means is that there's so many variables that impact the coverage that we are getting. So it's not just enough for us to look at the physiology and the toxicology of our pesticides. We also need to look at the engineering, at the how to spray, how to optimize the spray. So, and what has led me to research this further is, of course, these yellow cards, these water sensitive spray cards that are available. You can use them to quantify the spray coverage. And I will just point out that this is me standing here when I do commercial industry trials. I'm doing all the spraying, so I know what I'm talking about. And I also know what it's like in July to turn off the fans and have to apply 15 different treatments. And so run the different chemicals and rinse your tank and all that 3 times in between. I know all of that. So we know the reality. We know how important or how likely it is, how dangerous it is. We know how easy it is that people are going to be cutting corners, because this is very, very, not very desirable. So we need to come up with better ways. We need to automate it. We need to think of ways to, not only in field crops, but also inside greenhouses, how can we make this better? So what we have done is we have developed a roboticized system. So here we are. I've sort of slowed it down a little bit. So we have a rail system where we actually run 3 nozzles on a commercial setup where we can change the nozzle spacing, the boom height, the pressure, the nozzles. We can simulate whatever commercial settings you want. And we can do that and repeatedly do something that actually simulates what is being done either in a greenhouse or in a field setting. And then we have also published and made freely available a phone app. So here I went together with some computer science students, and they did all the coding for Android and for iOS. And you can now download Smart Spray to actually quantify this spray coverage. So if you are working with pesticides, I strongly encourage you to, if you're not already doing it, use these water-sensitive spray cards, and then use technologies like Smart Spray to figure out what kind of coverage you're getting and get an idea of how consistent your spray coverage is when these chemicals are applied. And then we can take that, we can of course improve our production systems, but also from an academic standpoint, there's so much we can learn about this if we just broadly talk about bioresponse as a function of spray coverage. There's so many ways that we can take this system where we can regulate spray coverage, and we can ask all sorts of questions, such as behavioral avoidance, where we can basically see when we give pests a choice between 100% coverage and 0% coverage or 50% coverage, we can see that they shy away. That the pest, one of the reasons why we have such huge problems with pests is, and why we have this evolution of pesticide resistance, has to do with them being exposed to sublethal effects. So we need to make sure that we get high spray coverage. All of that can be characterized, can be modeled, can be experimentally tested with something like this. So we can really get into the behavioral ecology and use pesticide sprays as a way to study this. We don't need to go to Madagascar or to Seychelles or something. We can go to a strawberry field in Salinas, and we can do very, very interesting and very interesting behavioral ecology, in my view. We can look at the sublethal effects of pesticides, and of course combine all the different technologies and disciplines that we are aware of, rooting it into this work with pesticides that we can experimentally control using our roboticized system. And of course, if we also get the economists and the sort of system agronomists involved, we can work with them. So this really opens up a lot of doors for us to work with industry, for us to write grants that are different, because we have this technology now, and can really simulate and validate lab experiments. So we're very proud of this system. And the guy who developed this system is on the next slide here. This is Anil. He also helps, so he's doing all the robotics in my department. What you're looking at here is then the fourth pillar that I'm going to be talking about, and that is our optical sensing, which is sort of where I do most of my work myself. So we have developed a system. This is a hyperspectral camera that can also run on the same rail system, just like the spray rig, but we can also put this hyperspectral camera on a drone, so we can fly it. We fly over fields. We can put it inside a greenhouse on a rail system, and we can also do this where we actually put it inside, and we have a control system where the system basically scans a certain level of some crops that we have experimentally infested in our greenhouse or subjected to different growth promoters or whatever classes we have experimentally subjected them to or treatments. We can then bring them into a room, and we can then collect very detailed spectral data and analyze that data. And I know that there are people in the audience that work in this space, so this might be of interest to you. So what you see here is a typical reflectance profile of a green plant, where it has low reflectance in the blue and in the red and more in the green, and then there's this red edge, and then it gets high up in the near infrared. So you can have plants and you can look at those profiles. Well, there's another way of presenting this, and that is where you sort of divide one with the other, and the black horizontal line will then imply that there's no difference between the two, that the reflectance values in the two plants are the same, while in the areas where there's an upregulation of reflectance, well, that's maybe where you have your responses. So those of you who work with genes, you're looking at upregulation of genes, or if you work with volatiles, you're looking at volatile profiles and see which volatiles are going up and which ones are going down. We're doing the exact same thing, but we're working with reflectance values in spectral bands, okay? So we can do that, and with the optical sensing system that we have set up, we are posing a question which I find extraordinary that nobody else has asked, because I have looked into the literature and I have not found anything. And that is this simple question of, if you're collecting reflectance data from plants, should you do it during the day or should you do it at night? There's no study out there. We now have the data and we are in the process of writing this up. So let me just show you. So we took a whole bunch of plants and we imaged them at day, right around noon, and then also at midnight, okay? And if you look at it, so now dividing day with night, right? So night with day, you can see that there's actually considerable difference. There's some area there between 500 and 600 nanometers, and then again around 700 nanometers, where there's a dramatic change, a shift in difference between day and night reflectance. And then what we have as part of our treatment was that some of the plants were infested with spider mites and some of them were not. So the real question we are interested in was, can spider mite-induced stress response be detected more accurately during the day or during the night? And the answer is, at this point, it's during the night. That you can see the blue here is there's a much bigger response to spider mites at night than there is during the day. Now, this is very, very important from an operational standpoint, because that means that you can run this at night where you don't have this annoying sun creating light that confuses your signal. So you can use artificial lighting. You can work inside greenhouses when nobody else is around. So you can have your rover or your rail system or whatnot operate when there's nobody else in the greenhouse, okay? Collect the data, send them to a supercomputer, have them analyzed, and then you show up next day, and then you have your results, right? That would be the dream, okay? But so that's really interesting to know that we actually have this potentially as a way to do this more effectively. And then finally, what I want to talk about is we can also apply this technology. So we can fly it on a drone, we can put it on a rail system, but we can also do something here where we take the exact same camera and we now put it above a hemisphere that we have coated on the inside with something that is very reflective. And then we have some objects on a conveyor belt that are being moved underneath this hemisphere. And then we are imaging them. And what I'm talking about here in particular is crop seeds. We're very interested in looking at difference between dead and live seeds, seeds with and without viruses, male, female, diploid, triploid, all these characteristics that many of you are interested in as breeders, as commercial seed producers. There are many, many people out there that are very interested in knowing what is the proportion of my seeds that are dead or alive that have these different traits. Well, can reflectance data be used non-destructively to classify and analyze different seed samples? So what we are able to do is, as you can see here, so these are tomato seeds, right? So we have the tomato seeds running into the left. You can see them running in, and you can see the camera and the software. So this has taken a long time. This is not something I have developed, but this is something we've had people develop, where you actually take those seeds and as they are being imaged, they are being individually saved as individual seeds that have a number that it goes into a file. So if we collect these seeds, we will know the reflectance profile from each of these seeds, okay? So we can then afterwards import this directly into statistical packages and run all sorts of fancy analysis of this. And if we do that, and here's an example of what we're capable of doing. So here we have, again, the same sample 1 divided with sample 2. So the graph to the left shows you the difference. You can see that there's more than 30% difference in some of the bands. So these are very different samples. Well, when we then run some basic analysis, we can also see that the seeds separate out very, very easily. So no surprise there. If we have very, very little difference, so it's close to 1, we can also see that there's very little difference. Now, this may be something you're interested in if you're trying to look for outliers. But this could also be this could be actually a very valuable result for you if you're into production and you want to show consistency over time. If you have seeds that were grown from different locations, you want them to be consistent year after year or from different places, this analysis could be used for that. And we can do that very, very quickly. And then if we take it even further, we can also get into something where we actually have then, as the seeds are being analyzed, we then also real time have them analyzed, so you can actually have them separated out. And then you could have these air jets running on the downstream end of your conveyor belt and have them shoot into different bins. So you just put your seeds into a hopper, you go to bed, or you watch some football or something, and then you come back, and then the seeds are all separated out for you, okay? So, in summary, what I hope I have shared with you is that I think that insect ecology and pest management is sexy. I think we have an opportunity here. I think we are underselling a very valuable product. I think we have a big job to do to get the message out and expose more people from computer science, from engineering, from anthropology, so here Manshu is with me today. She's from arts department. She's helping making a video out of this. So we are also making extension videos, and we have them translated into Spanish. So there are all sorts of ways that we can use this platform to recruit students who have all sorts of skill sets, and really look at the challenges, like I said, as opportunities. How we can take these technologies and really help these young, fine people create great careers, and really embrace the diversity of academic experience and put it together in teams that can produce cool stuff. So, because students and society expect us to do that, and I think we need to take that responsibility very, very seriously. So we are really into trying to combine insect ecology, engineering and programming as something that we do a lot in my program. And really also, ultimately, we want to make money. I want my students to go out and start businesses. I want them to get involved in entrepreneurship, not just write papers about it, but actually take it all the way out. And with that, I want to thank you, everybody, and enjoy your dinner.